This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 5, recorded on April 15th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Today, we're recording on Tax Day in the U.S., although it's not actually due today anymore. Joining me today from the Medical College of South Carolina, Michael Schmidt.
Hello, Vincent. How are you this afternoon?
Very well. Beautiful day here in New York City.
Outstanding.
Blue skies, 55 degrees. Couldn't ask for more. Also joining us today from Princeton, New Jersey, Cliff Mintz. Hey, Cliff.
Good afternoon, Vincent.
How are you on this tax day?
I finally submitted my taxes, even though I didn't realize that we had until the 18th. I figured, what the heck? Well,
when I learned the other night that we had a few more days, I just stopped working on it. Leave it to the last minute. That's my motto. Also joining us today from the University of Louisville, Ron Atlas. Hey, Ron.
Hey, Vince. How are you?
You have nice weather there as well?
No, we're getting this wonderful storm that you're going to get tomorrow, although we're not getting the tornadoes, fortunately.
Yeah, it was really bad. Where was it? Arkansas, Oklahoma? Oklahoma was awful. 6 people died so far.
Yep.
Well, I'm sorry that we're going to get rain and wind tomorrow, but keep me inside to do my taxes. That's all right. Well, today we have a couple of interesting stories to talk about. And the first one, actually, both of them I culled from microbeworld.org. So to our listeners, check out the site. If you like a story, tag it with TWiM. Just go to a story, and then there's a little box where you can write TWiM in it, and it tags it. And that way I can see what you guys want to hear. Otherwise, we just keep picking the stories ourselves because there's no shortage of good stuff to talk about with respect to microbes. Our first story is the genome sequence of a Mercury-methylating Desulfovibrio strain. And this came out in a journal that I used to read all the time as a graduate student, the Journal of Bacteriology. And I have to admit, in recent years, I haven't read as much. But this is a very short paper. It's less than 2 pages, but of course all the data that have resulted from it are quite substantial. This is a bacterium with a 3.8 million base pair genome that encodes 3,455 candidate protein coding genes. How does that compare to other bacterial genomes? Anyone know offhand in size?
Well, the rule of thumb, I always try to impart my wisdom to the medical students when they ask me these fact-based questions. And I said the rule of thumb is about 4,000. I always compare the complexity of the microorganism, which can do wonders in the human body, causing infection and providing us with all our vitamins, to that of a 747, which has millions of the equivalent of genes moving about, and yet it cannot replicate itself and it cannot do all the wonder that the bacteria do. So I would say that this is a garden variety genome for the microbial world, especially since this is coming from, depending on your perspective, either a very old niche or a very specialized niche. Right. The other remarkable thing about this particular manuscript, and it being so short, is I think it speaks volumes about how hard it is to talk today about genomes and the tools that are going to have to be developed for us to be able to image and think about the genetic complexity associated with, in this particular case, the methylation of mercury and the environmental consequences associated with the volatilization of methyl forms of mercury moving through the atmosphere and then effectively raining down in various locations throughout the country, depending on where it rains.
I guess, Vince, in answer to your question about how big it is, it's about 4 times, I guess, bigger than the first bacterial genome that was sequenced. It's half as large as one that I was involved with, which was a Nostoc strains. In today's sequencing, it's a big job, but it's not a monumental job anymore to get a bacterial genome of this size totally sequenced.
Right. So the interest here, of course, is that this bacterium, and in fact, it's a specific one. It's Disulfovibrio desulfuricans strain ND132. It's an anaerobic sulfate-reducing bacterium that can produce methyl mercury from mercury, of course. So the idea is that they would like to figure out how this bacterium does this. And much of this is new to me. I didn't realize that these bacteria actually did this, that they actually took mercury, which we supply largely into the environment with burning fossil fuels, and they convert it to methyl mercury, which is quite toxic to humans. Listeners may remember the Minamata, Japan incident a number of years ago when methyl mercury was being dumped into the ocean. It gets concentrated in the food chain, and the fish eventually have lots of it. People eat the fish. And many, many kids were born with developmental defects until the company stopped dumping it into the water. I understand we don't dump methyl mercury in the environment that much anymore. But Dixon de Pommier told me they used to use it to bleach wood pulp that was used for paper, and then they would throw it away after using it. But of course, we make a lot of mercury, and that's what these bacteria do. So I wondered right off the bat, why does a bacterium take up mercury and convert it to methyl mercury?
Well, there's 2 reasons for it. The first you've already alluded to, it's toxic. And microbes, not being fools, recognize the easiest way to get rid of a toxic product or a waste product is to volatilize it. So the methyl mercury will be volatile from the perspective of the microbe, so it'll go off. And the second reason is all microbes need to find a place to place their waste. And since, as you pointed out, that this is an anaerobe, it is going to run out of a place to put its waste electrons. And the good place to put waste electrons is into something like mercury, where it can then reduce the mercury by giving it the electrons, and it can go off.
And so the reduced mercury then combines with a methyl group at some point.
Yes.
Is that the idea?
That's the idea, and it goes off as a gas.
So I noticed in the paper they said that there are known bacterial genes that confer mercury resistance, but they were not present in this bacterial genome. So that's consistent with what you're saying, Michael, that it would not otherwise be toxic, the mercury, to these bacteria, and therefore they have to convert it to something that to them is not toxic.
And they dump their waste at the same time. It's a twofer.
So why isn't methyl mercury toxic to Disulfovibrio? Do we understand that?
It's principally because they're coughing up the electrons to it. The methyl mercury is leaving the system, going off as a volatile. Mm-hmm. It's leaving the system.
I see. And in us, methyl mercury, which is actually a cation, I guess, it combines readily with anions, especially those with sulfur, like cysteine-containing proteins. So it binds our proteins and has its toxic effects that way, I believe. These bacteria, if I understand, typically live in aquatic environments. They're often present in mud or silt areas, and that's where they pick up the mercury and convert it to methyl mercury. We make 80 tons of mercury a year in the U.S. released into the atmosphere. It's a lot.
And where does it come from?
My understanding is that's largely from burning fossil fuels.
That's absolutely correct.
So we have a lot of coal-burning plants still. Automobiles, I presume, right?
Well, fortunately, we've gotten most of the nasties, or we tried to get most of the nasties out of the fossil fuel. We got rid of the lead, and we're continuing, hopefully, to get rid of the other forms of toxic compounds as best we can out of the mercury.
Now, Ron, when I was reading about these bacteria, in one of the papers it said that they're often used for bioremediation because they can reduce uranium, chromium, and iron. So I'm sure you must know something about that.
Yeah, it's an interesting group of bacteria, and they do, in fact, reduce a number of metals. So mercury is just one of the metals that's involved, but uranium and others. Some of the strains also dechlorinate compounds. So in many ways, they have a real high potential for bioremediation of areas that are contaminated with chlorinated compounds and heavy metals.
So you say potential. They haven't actually been used in that way yet?
I'm not aware of any large-scale use, but certainly the Department of Energy has been looking at any number of ways of cleaning up sites that they've managed to contaminate.
So by sequencing the genome, how will they find out what genes are involved in methylation of mercury?
That's going to be an interesting question. I guess the surprise of this paper for some of us is that the mercury resistance genes that you find in some other bacteria are in fact absent in this particular bacterium. So it's a different mechanism of resistance to mercury than has previously been reported. I was fully expecting when I saw the paper that they were going to say, and we've identified the mercury-resistant genes. They went just the opposite way.
So would you expect the resistance have to overlap with the methylmercury production genes then?
Yeah, I expect resistance to be due to methylation of the mercury. That's the theory that this organism, as Michael was just explaining, will take mercury, convert it to methylmercury, and thus the bacterium lives happily, even if it gets into animals and humans and causes problems otherwise.
So what this means is that there must be novel genes that we're not aware of that carry out this process in this bacterium.
Yeah, and I guess that is the real ongoing value of the full genome sequencing. We've looked at well over a decade of study of well-characterized genes for methylation of mercury, and it's just not in this bacterium.
I think with this particular paper and where this discussion is going is really akin to what happened about 10 to 15 years ago when we had the emergence of the concept of pathogenicity islands, these regions of DNA first discovered in gram-negative bacteria where there was novel sequence, oftentimes very large regions of the genome that were transited into the bacterial genome that conferred special properties to that particular organism. So one way we might begin to go hunting for the mercury resistance genes is to compare two species of Disulfovibrio, one that is able to deal with methylating mercury and one that is unable to deal with the methylation of mercury, and then literally compare and contrast their whole genomes. Fortunately, today we have much more computational power than we did 15 to 20 years ago when pathogenicity islands were being described. And consequently, I think it will be a much easier task for this group of authors to go out to their favorite source of Disulfovibrio, isolate some microbes that will not methylate the mercury, and then literally extract the genome, run it through a 454 process where you can get it. And since you have a draft-ready sequence already, you can then easily compare and contrast those sequences and tumble into where these redox proteins are that have the ability to methylate the mercury. And I think we may discover some new resistance mechanisms potentially. We may discover some new transport proteins that are able to move the mercury in and out of the cell very easily. We may even discover some new type of cytochrome or iron sulfur proteins that have a predilection for these heavy metals that are involved in redox processes. Because from the microbe's perspective, it's really about redox. And so I think what we may see come out of work like this is the concept of redox islands, akin to what pathogenicity islands are, or if you will, environmental islands that confer a selective advantage to a microbe to live in a particular niche that has a particularly interesting molecule from which it has to have metabolic challenges or toxicity challenges. Because microbes are really simple creatures. To them, it's all about redox. Because if you go back to the simplest example of taking glucose and converting it to lactic acid, the lactic acid bacteria spend their entire career effectively taking pyruvate and converting it to lactic acid, not because they like living in acid, simply because they have no other mechanism to generate oxidized NAD. And you can't eat 1 mole of glucose unless you have oxidized NAD to accept that initial pair of electrons in the burn when you begin to split glucose into its subsequent smaller 3-carbon units. So from the microbe's perspective, it's all about energy and where your electron donor is, and then how you process those waste electrons to regenerate that initial electron acceptor that enables you to use these materials as energy sources or electron sinks, depending on which side of the equation you're on.
So probably we should explain this a bit, because I'm sure there's some listeners that don't remember their electron transport. So basically you start an electron transport chain to generate a gradient of electrons, either in eukaryotic cells or in bacteria. And that gradient is then used by enzymes to produce energy in the form of ATP. Is that a fair?
That's a fair statement. It's all about flux. It's about those flow of electrons.
And in us, we use the last electron acceptor is oxygen, right?
Correct.
Right, and in the anaerobic respiration,
right,
which these guys are, there's always whatever final electron acceptor is besides oxygen.
Right,
doing anaerobic respiration is what they're going to use to accept the electron.
So I understand that these guys can use the Disulfovibrio use sulfur quite frequently.
Right. So it depends. So what Michael was saying is correct. It's kind of like what's ever available in whatever environment they find themselves in, they could use whatever final electron acceptor is there so that they could accomplish their anaerobic respiration.
So that includes mercury?
I believe so.
So, Michael, is that the terminal, one of the terminal electron acceptors?
Any port in the storm that you can dump waste into, microbes will take advantage of.
Great.
But that's why Vincent, when you look at contaminated soils,
right,
and no matter what's there, frequently you isolate a bacterium. A lot of them are archaebacterium,
right?
But they're there, and like there are methane oxidizing bacteria, and then there are methane producing bacteria.
Right.
Now, it's interesting that you mentioned that, because originally the thought was that it was the methanogenic organisms that were in fact methylating mercury, and everyone was looking at that, and eventually there was an experiment that added molybdate to the sediments that were evolving methyl mercury, and it stopped evolving methyl mercury. Now, molybdate was known to inhibit the sulfate-reducing bacteria, and that's really what led to the discovery that the Disulfalvibrios were involved in the methylation, and it wasn't coming from the methanogens. What is known is it's a cobalamin-linked reaction, but there's not nearly as much known about going from mercury to methyl mercury as there is about going from methyl mercury to dimethyl mercury. This is a much more complex sort of cycling reaction where some organisms under anaerobic conditions are forming methyl and dimethyl mercury, Others under oxidative systems are going the reverse way. So there is a cycling reaction involving mercury. I guess I'm not sure that this is actually involved in any energy generating reactions, but it certainly seems to be a detoxification reaction for the bacteria.
I see. Okay. So are there any other bacteria that methylate mercury, or is this the only known one?
No, I'm sure there are other bacteria that methylate mercury. And I should know what they are, but for the moment they're skipping my mind.
Okay.
It's a safe bet to bet on the positive of that.
Right.
Yes, I would say this. So how does this fit into for the people that are not microbial physiologists? I mean, what is the role of bacteria from a global perspective into the geochemical cycle? Because I don't think people really know how much contribution there is from microorganisms to, keep the geochemical cycle going.
Well, I guess the answer I always give my classes is if we didn't have these geochemical cycles that were all based in the microbial world, all life on Earth ceases within 2 weeks. Mm-hmm. So we rely upon these microbes to keep cycling substances and sending some out to the atmosphere and bringing some back into the aquatic and soil ecosystems.
So these include the nitrogen cycle, right? The carbon cycle.
Nitrogen cycle, and in this bacterial case, it's the sulfur cycle.
Right.
Because these are sulfate-reducing bacteria that produce hydrogen sulfide. There are other thiobacilli and others that take the hydrogen sulfide and bring it back into sulfate. And you can sustain those cycles as long as you have different groups of bacteria, some that are in anaerobic systems like this one, pumping things out, and others in aerobic systems. And up and back, substances go.
I just think that it's very important for people who listen to TWiM to understand that while the pathogenic organisms or bacteria get a lot of the press coverage, the majority of bacteria that are around us are very beneficial to humans. And I think that the public at large has a tendency to equate bacteria with just bad things like disease, but in fact, it's only a very small percentage of the kingdom or the overall bacterial kingdom that does bad things to humans. So it's just important.
And thank goodness for that, because there are an awful lot of bacteria out there.
Just in us, there are tons, right? More than cells that we have in our bodies.
But it's important to get the word out that bacteria are good, not just bad.
Absolutely. Microbes, we need them. I have bumper stickers all over the place from ASM that say that. How many bacterial cells do we have in us?
It's about 100-fold greater than what we have in our own human cells.
Yeah, it's like 100
Unless
trillion,
you count the mitochondria inside our cells, and then all the numbers go really crazy.
Is it 10 to the 12th? I don't even know.
Well, we can't be saying this. Let's see. I'm going to Google it right now. How many bacteria in humans?
The average human.
The average human. Why some have more than others, right, Cliff?
Well, it depends on your size, I guess, right? Surface area.
Oh, look, a little quote by Margaret McFall Nye. Well, she said this was 2008. That's bound to be old. For some reason, I have 100 trillion in my head.
That sounds about right.
And we have a couple of trillion human cells in us, I think, if I'm not mistaken.
Well, Vince, your assignment for the week is to go count them.
I've counted 500 to 1,000 species in the gut alone.
Yes.
10 to the 14th bacteria, 10 to the 13th human cells.
Thought last week that there were even more viruses in our bodies.
Absolutely. Yes, that's just starting those kinds of studies. A few people have done some human virome analyses, and Jeff Gordon at WashU, who has been looking at the gut microbiome involved in obesity, has found many, many viruses in there. This is going to be very interesting to sort out. If I were just starting out, that's what I would head for.
That and ways to visualize all this genomic data that are coming out. I think the people who can begin to think about how to reduce the A's, C's, and T's to intelligible figures and diagrams so that those of us who were taught to think microbially would be able to make some sense out of it without having advanced degrees in linear algebra.
That's right.
I know.
You know what I find interesting about this approach here? Michael, you said this before. You get two bacteria, one that methylates mercury and the other that doesn't, and you compare their sequences. When I was a graduate student, if you proposed that kind of experiment to answer a question, you'd fail your qualifier exams.
Absolutely.
Because you have to do genetics to answer that, or biochemistry.
Well, we were all taught to be able to. We were all trained as pure culture microbiologists,
Right.
and I think only those individuals.
Speak for yourself, Michael.
Oh, excuse me, present company. You were then trained in the Hungate School. Where
I went off into places no one else would go.
Yes,
that's good, but you probably wouldn't advocate comparing whole genome sequences, would you? Or maybe you would, Ron.
I would.
Yeah. I'm not saying it's bad. It's just that it's evolved to this now because we can do it so readily. Back when I was a student, you could sequence one or two genes, and it was usually better to design a genetic or biochemical experiment.
But to give the microbial physiologists and the pure culture people, or the people that were trained as cultural microbiologists, is that the lament is that every student that comes out of a graduate program really knows how to do molecular biology well, but they don't really know bacteriology or microbiology very well anymore.
Yeah.
Which is, there's an upside and downside to everything, and there's always balance. So I think that, but we shifted recently to molecular because that's where the action is.
Well, some people argue that in the future you'll just do virtual experiments and look at the sequences.
No, I think the microbes are going to continue to surprise us, because the issue is, even with our advanced computing power of today, with 5,000 different species all interacting with us in our gut, you have 5,000 species all interacting with our human genome at the same time. And each and every one of those microbes within that 5,000 species community is actually sending out signals that we react to and they react to, and it's a constant iterative process. And so it gets extremely complex trying to interpret it. So you're going to have to be able to go back and do very sophisticated addition experiments where you add back so many players to the mix, right, in order to ask what's going on. It goes back to the gnotobiotic animals, in which you're effectively born germ-free without any microbial flora whatsoever, and then you add back a baseline flora to the whole animal, and then you begin to ask questions about hormonal signaling as well as the other things. And I think the environmental isolates, since we're talking today about the environment affords us to do those sorts of add back experiments in a much easier and less complex way than doing it in human trials or animal trials, where the complexity is so much greater. So I think papers like this, where we're talking about a straightforward process of methylating something like mercury in bottles, will be much more easy to do, where you can add many different variations of experiments in which you can do old-time physiology experiments asking what's going on, and most importantly, you will be able to do mass balance, because I think it's only through mass balance, where you measure your input and your output, that you're actually going to be able to decipher some of these complex interactions that are going on amongst communities.
Right. I wanted to ask if the methylmercury produced by Desulfovibrio, does that constitute a threat to humans because it gets concentrated in the aquatic food chains? Does anyone know?
Yeah, I mean, it would seem to be that if we add sufficient amount of mercury to the environment, and there are anaerobic environments, that this then produces enough methylmercury to get into fish, which then, if eaten by people, are a threat.
Okay. So now if we find the genes involved in this bacterium someday, what can we do with that? There's no way we can alter the production by these bacteria, right?
Not on a global scale.
Yeah.
You may be able to develop bioreactors that may be able to harvest the mercury, and if you have a particularly contaminated site, you may be able to develop a scrubber-like technology, engineering the microbe so that rather than volatilizing it, it will fall out of solution. You could try, or you could engineer it so that they have different endpoints or are able to accumulate, or take it to an elemental form where it falls out of solution and is not bioavailable.
Maybe we should just clean up the fossil fuel burning.
That's an alternative too.
There's a novel idea.
Well, we're going to run out of it eventually, right?
Yeah.
So then we won't have any mercury. Starting at the source sometimes helps. Well, I don't know too much about this, but it seems to me that, as Ron said, you probably can't change the bacteria, but you would learn how they dealt with mercury and perhaps could use that in some way. But it's not something that's discussed at all in the paper, of course, being less than 2 pages.
Yeah, it's an amazingly short paper,
Yeah.
and it's amazingly focused on one metabolic activity, considering that they have now sequenced a whole genome of a bacterium that's major importance in ecology is sulfate reduction.
That's a next paper.
Well, I would have thought they would have discussed this somewhat. Usually when you sequence a genome, you talk about what you find, what's new, and where it might lead, but they haven't done this at all. It's quite unusual.
I was surprised too.
Yeah. Nevertheless, as you can see, it does generate discussion.
Well, more than that, between this morning and the time we just started this discussion, this has hit the popular press. And so if you Google this topic, it's just suddenly lighting up with one article after another about it. But no one has said much in the press about what it really means.
That's because they don't know.
Yeah, they don't. Right? I think they know less than we do.
I think it's difficult to understand, bacterial physiology or microbial physiology and make sense of it in the popular press.
Yeah. Well, so this episode will release at just the right time.
Right.
Good. Well, I thought it was very interesting. I thought it was very interesting, and I'd known very little. So there you go. All right, that's our first story. The second also has to do with microbes in the environment, and this one is called Perturbation of an Arctic Soil Microbial Community by Metal Nanoparticles. Here's another area that is totally new to me. Nanoparticles apparently are everywhere. They're things that we make. They're in electronics, foods, clothing, medical devices. We use them for experiments in the laboratory. And they're very small particles, 100 nanometers or so in size. They're made of various metals, silver, copper, even silica. And as we make more of them, they enter the environment because they blow around on the winds. So in this paper, they took some soils from the Arctic, from an island almost as far north as you can go in Canada. And this soil was assumed not to have nanoparticles in it yet, anyway. Hasn't reached there yet. They did an analysis of the microbial community of this soil in a variety of ways. Then they added some silver nanoparticles to this soil and let it sit for a while, and then looked at the effect on the microbial communities. And what they found was that the silver nanoparticles are quite toxic. And in particular, one of the bacteria associated with plants, normally a nitrogen-fixing bacteria, Bradyrhizobium, is particularly sensitive to silver nanoparticles. So who knows about nanoparticles?
I know the United States EPA is very much interested in toxicity associated with nanoparticles for all the obvious reason. They're unknown, and there's no substantial body of literature that argues one way or the other whether or not they are going to have long-term consequences for people working with them. With that said, I think their ability to move easily within the environment because of their size. Remember, these nanoparticles are smaller than bacteria. They're 20 nanometers in size on average is what these authors used in their particular paper, which make them smaller than microbes. And one of the things that we do know about microbes is that they can remain suspended in an air column for a long period of time. So if you're working with them, they're often worked with in clean rooms or in hoods to control their spread. The standard N95 mask that many individuals wear to prevent microbial contamination often would not work to the level and extent that N95 masks are used to protect you as an individual from microbial attack because the particles are so small, just as N95 masks have limited utility for controlling viral attacks.
Right.
And so consequently, I think that was interesting. The other aspect about this particular paper is they were looking at compounds that are known to be antimicrobial. Silver has been known for quite some time to have an antimicrobial property associated with it. After all, silver nitrate drops were some of the first antimicrobials that many of us were exposed to when we were born before the development of tetracycline-based ointments or drops that were placed in infants' eyes as they were born to control the spread of chlamydia trachomatis, which is the bacterium that would often be implicated in blindness. And so many of us, when we were born by public health law, actually had silver nitrate drops placed in our eyes to inactivate those microorganisms that we may have acquired should our mother have been colonized with that variant of chlamydia. So silver has been known for quite some time, but I think what's remarkable here and in this paper is the size of the particles and how they interact with the soil. And in anything that has an antimicrobial property, it's all about availability. And these nanospheres, because what they demonstrate here in this case of the soil, were, if you will, bioavailable to the community because we saw a reduction in the number of microorganisms associated with this complex soil.
Mm-hmm.
And so thinking about it from a medical perspective of being able to use something like nanospheres to control infections, especially those that are chronic infections, biofilm-based infections, this may actually suggest an application for these if, again, we could figure out how to deliver them to their biofilm. So looking at these silver nanospheres, it was interesting how effective they were, and the only microbes that seemed to survive pretty well were the ones that formed spores. And for better or worse, spores aren't really alive, if you will. They're just pluripotent bacteria waiting for the right circumstance that will enable them to germinate.
They actually mixed the soil, the Arctic soil, with silver nanoparticles and incubated for 176 days at room temperature. I guess I don't know how they arrived at that number, but I guess it's to simulate nanoparticles coming out of the atmosphere, sitting in the soil, and then being there for long periods of time. And then they, as Michael said, they sampled the communities. They used a number of interesting ways to do this. They looked at metabolism in the soil, and they analyzed the metabolites as a way of looking at the change in the communities. And that's something that was new to me. Is this a common way of looking at microbial communities?
Yeah, it is. I mean, this is sort of a very general approach to looking at microbial ecology and how various substances impact those communities. Today, we also look at it by doing genomic and metagenomic analyses.
Right.
And Bradyrhizobium, which is the bacterium they found the greatest effect with, is a particularly sensitive organism. In fact, the microbes involved in the nitrogen cycle sometimes are used as indicators of environmental stress because they are particularly sensitive.
Mm-hmm.
One of the neat techniques that they used, Vincent, was one that Ron has used when he was studying the oil spill of 20 plus years ago up in Alaska.
Uh-huh.
And that's the magic most probable number using PCR. And Ron, do you want to expand on the wonder that PCR-based MPN is all about?
Well, we certainly use most probable number methods as a statistical treatment for enumerating microorganisms, and then we're able to amplify particular organisms with PCR techniques and come up with estimates for numbers. It's a technique that's not often used, but it is out there for those who want to use it.
So what exactly is the most probable number?
We run a series of replicate dilutions to a point where you don't detect something, and then you do the number of detects in wells at that particular dilution versus non-detects, and there are statistical tables depending on how many replicates you run that allow you then to come up with an estimate of how many organisms or genes were in the original sample. So 3-tube and 10-tube dilutions are often run. It's a lot of work and a lot of replication as a rule, but it does give you a statistical estimate and some confidence limits.
What's really neat in this paper, it says, compared to control cultures, the presence of silver nanoparticles reduced the growth of this one particular organism by 8 orders of magnitude. And they, again, used mean MPNs were 1.3 times 10 to the 8 cells per mil versus 6. So this technique will give you, to a statistical certainty, the ability to detect 6 microbes per mil from a soil that had been incubated with silver nanoparticles for 170 days. I mean, that's what's truly magical about being able to say something to that level of certainty. You know, effectively, 1.3 times 10 to the 8th is 103 million bacteria versus 6.
It's impressive.
You know, what would be interesting for someone to look at is, since nanoparticles do distribute through the air, is what the impact on Staphylococci on the human skin would be. In burn victims, we used to use silver-containing ointments for controlling staph infection. So if you're worried about Arctic bacterial communities by aerosol distribution of silver-containing nanoparticles, one ought to be asking, what about staph in human skin?
Can you do that kind of experiment?
Michael can.
It's one of the fun facts for future figuring is you have to remember the average human is dropping 2 million skin cells an hour. Just sitting here, we're going to drop over 2 million skin cells times the 4 of us. So you look at how many skin cells are falling off, and they're typically covered with our normal flora, which for the most part of humans is coag-negative staphylococci. And so you can quickly sweep up the debris under your chair and incubate the dead skin along with copper nanoparticles, or excuse me, silver nanoparticles, to effectively ask that question, how good are these silver particles in terms of degrading the inherent microbial load? Because in the infectious world, or where we're worried about colonization of people with bad actors like MRSA or methicillin-resistant Staph aureus, you worry about how to effectively decolonize someone. So you could effectively cover their nose and hose them down with some nanospheres and ask the question, can you debride someone of these bad actors that may be inherently colonizing? And some of the colonization rates for some of the things like MRSA are approaching 30% in some populations of individuals working in healthcare institutions, as well as the general public.
You know, you mentioned, Michael, that I sometimes work in oil spills and that humans tend to slough skin. There's an interesting report out recently on the Gulf of Mexico following the BP oil spill that shows the disappearance of Propionibacterium acnes from marine waters. And it seems to be because people stopped swimming during the oil spill along the beaches. And so they stopped sloughing off their skin with Propionibacterium acnes.
Amazing.
I mean, I was astounded to see that you could actually make those measurements. Those were done using metagenomics.
Wow. That's impressive.
I was surprised that anybody would even think to consider asking the question.
Well, if you look at the total microbial communities, I guess you'd pick it up, right?
You just picked it up. I don't think it was a question someone asked, but it was still a surprise to me that you'd find it.
I guess you're right.
And some enterprising young attorney is probably going to use that fun fact to make the argument for the business case as to how much that oil spill impacted on tourism.
You may be right.
That's good data.
It is.
It is good data. And again, this really illustrates the power of metagenomics, of how you can perform a survey and to a statistical certainty argue that you had fewer swimmers because you saw fewer Propionibacteria acne microbes in the water. That's really cool, Ron. I hadn't heard that.
That's out of the University of Houston, and it's out in a Nature publication.
So now, in fact, in the discussion, they say when we started these experiments, we were skeptical about the toxicity of nanoparticles to microbes, and now we are really surprised to see this. So what do you do? I mean, you're not going to stop the production of nanoparticles. You make millions of tons a year. It would seem to me that they're not only a concern for Arctic communities, but microbial communities all over if they're so important for us, right? So what do we do next?
My guess is we're going to have to somehow monitor their release because, again, this is why I think the United States EPA is very much concerned about nanoparticles in the environment or nanoparticles in the workplace, in that we don't know what we don't know. And the ability to make something this small has only recently come into generalized manufacturing. And so we don't have a large volume of data on the consequences of these things moving through the environment in terms of what it may do to the microbial geochemical cycling. If we're going to argue about soils being very important in balancing many of the essential processes that is the planet. So I think studies like these are extremely important, and they have to be appropriately controlled and interpreted to understand what is actually happening in order to make an informed choice about how best to regulate the dispersion of nanoparticles or somehow controlling whether or not they get out into the environment.
It'll also be interesting as we start to see therapeutic drug delivery using nanoparticles, how many years before we start seeing any side effects from that.
Well, it's interesting that you bring that up, Ron, because I happened to run into a guy I went to graduate school with who is actually at the Food and Drug Administration, and he's in the toxicology division, and his primary area of research is nanoparticles and their use for therapeutic indications in humans. So there's a large the FDA is very concerned about what happens to these particles if they're going to be introduced into the body, where they accumulate, what they are actually going to do. And based on what he was telling me was that very few people that are developing these products are really paying attention to the toxicology of these things. There's no question that they work in the laboratory and that they're effective. The question is what happens when you use them in a clinical therapeutic setting. Will they actually harm or benefit a patient? And so I guess the interesting thing is that FDA is very aware of the emerging technology. And my sense from my friend who's 3 years from retirement is that he's not convinced that nanotechnology is the panacea that many people are betting on that it will be. So
one of the problems is that you can alter your own microbial communities with these nanoparticles, right?
Yes.
And that could be detrimental because we've just finished talking about how good they are, and we'll put these nanoparticles in us, it's going to change it. But as with any new technology, we don't really worry too much about the safety, right? We just go forward.
Well, that was his point. That was his point. From a scientific standpoint, they're fascinating, but their goal at the FDA is that in order for it to be useful for treating patients, it needs to be safe.
Yeah. Now, as a way of wrapping this up, in the research for this story, I found a brand new paper in Nature Chemistry called Biodegradable Nanostructures with Selective Lysis of Microbial Membranes. And in this paper, they synthesize polymers, which, when placed in solution, essentially form nanoparticles. They form what are called micelles. And these have high affinity because of their charge for the negatively charged bacterial membranes, and they actually inhibit their growth. They did a number of studies with a variety of bacteria. They used Bacillus subtilis, Enterococcus faecalis, Staph aureus, and also MRSA, and a fungus Cryptococcus neoformans. They had, I think, a couple of different formulations, and they found that 2 of these polymers had a very strong effect against MRSA in the fungus, for example. So they have typical MIC curves here, minimal inhibitory concentrations, and they take some pictures of these bacteria and show that the cell walls and the membranes are damaged by these nanoparticles. They also put them in mice and show they have limited or no toxicity in the limited numbers of studies that they did. So they're very excited about the potential of developing these synthetic polymers as treatments for infections.
Vincent, what this reminds me of is the old story of prions and how prions become catalytic. And of course, as we all know, that prions are, thought to be the infectious model, if you will, of Alzheimer's disease and dementia. So one of the first things that I would worry about of these polymers having this profound antimicrobial potential is whether or not it would make something as beneficial to us as the human gut, because their spectrum of activity, unless it's specifically engineered only to take out MRSA, as opposed to the 5,000 other beneficial species in our gut that are supplying us with all the essential vitamins that we need and hormonal signaling that regulates our digestion, I would be extremely concerned about developing a wonderful antimicrobial that would take out a pest while at the same time causing our gut to become senile, and all of a sudden we're left without a way to properly digest our food.
Yeah.
I mean, from my perspective, Vincent, what they're trying to replicate are cationic antimicrobial peptides that are found throughout nature. So meganins are my favorites because they were found from frog skin originally. And there was actually a company way back in the 1980s called Meganins Inc., and they were trying to extract these antimicrobial peptides and use them therapeutically. And as they say in the article, these antimicrobial peptides that include meganins, defensins, which are found in human lysosomal granules, can be effective as antimicrobials. But the thing about this paper, and I have issues with this paper, somebody who's worked in this area, is that their chemistry is elegant, and it's clear that these guys are chemists. But when you look at their MICs, their data, you need to be well within the 0.1 microgram range up to maybe 5 to 10 at max for any kind of therapeutic benefit. And using these compounds, when you have a something that forms a micelle that's got a cationic or positive charge, they are known, it's been known for many, many years that these disrupt cell walls and cell membranes. And so while their compounds are interesting, they're not new or novel per se, and people have been trying to commercialize these peptides for the last 25 years. And as they say in their introduction, they're plagued by toxicity issues, rapid turnover, et cetera, et cetera. But most importantly, from this paper, the one experiment that they didn't do, which was really, really important as far as I'm concerned. I mean, they went out of their way and did toxicity, so they spent a lot of money on animals to show that they didn't, cause any damage or toxicological effects on the animals. But at the end of the day, the one thing that everybody who's antibacterial drug discovery does is you have to take your chemically synthesized compounds or your natural products and do your MICs in the presence and absence of serum, human serum. Because more often than not, because these particles are so charged and they're amphiphilic, even though they form micelles, they interact with other proteins. They'll interact with proteins, and if the proteins bind, or somehow it's usually albumin or some unknown other protein that's found in human serum, if they're sequestered by the proteins, they cannot be available to kill the bacteria.
Right.
So,
right.
and I actually consulted for a company that developed compounds similar to these, and it worked great in buffer, but once we added human serum and did titrations down to very small amounts of serum, they lost their antimicrobial activity. So I think the concept is great, but it's nothing new or novel. But it's great chemistry.
Maybe the chemistry is the novel part, right?
The chemistry is fantastic. I mean, it was beautiful chemistry.
So the serum experiment, maybe they did it and it didn't work, and they wanted to publish this.
Those. It's funny. It's funny you say that, Vincent, because I was hired as a consultant to help this other company that was looking for new antibacterials, and I kept on saying to them, look, before we try and get a deal with Merck or Pfizer or anybody, you got to do the experiments.
Sure.
And we were out looking for deals and stuff before they did the experiments, and invariably, every place we stopped, someone said, have you done the experiments?
Yeah.
And they finally did the experiments, and they did, the compounds no longer had antibacterial activity. So it was kind of a great deal for me because I got to fly all over the world and consult for an Australian company. But I knew that they were, it was not going to happen. So it's really important. I mean, the bottom line is that we are running out of antibiotics, and really everybody seems to be focused on methicillin-resistant Staph aureus and vancomycin-resistant enterococcus, and that's we do have drugs to treat those. What's really, really important that people are not talking about are gram-negative bacteria that there are no good drugs for, that do not cause pretty bad side effects for people. We have drugs to treat those infections, but those are drugs that physicians are reluctant to use unless there's no other option. And that's and you may have heard about the India strain with the new metallo beta lactamase inhibitor coming out of India. And I just had a conversation with somebody who's very much concerned about antibiotic resistance and bugs that cause, or bacteria that causes diseases. And he was telling me they've done experiments where the strain that carries this new, the gram-negative bacteria, Klebsiella and related gram-negatives, and this strain, they look for it everywhere, and it's found everywhere in the water, on the environment in India, which means that if it's everywhere, these E. coli, Klebsiella, whatever, are going to now be ingested by people and carried in a lot of people's gastrointestinal tracts, and these people travel. This thing started out, I think, 3 or 4 months ago, isolate only in India, and I think it's now found in 30 or 40 countries in the world. And the ID, the infectious disease clinicians, are really shaky in their boots, because after you can no longer use cephalosporins and penicillin-based antibiotics, there's really not a lot left. And the fear is that in addition to the metalloprotease, there's also additional antibiotic resistance genes that are found on the plasmids, the mobile genetic elements that transmit this toxin. Mm. So we're in for a rude awakening over the next 3 or 4 years. I
think we should do a show on this.
I agree, but the push towards using nanoparticles to deliver antibiotics or novel antibiotics I think is a very interesting field and ought to be explored further.
We also have nature's nanoparticles, and that's phage. And in the March issue of Microbe, they had a discussion of phage therapy,
Right.
which was nature's way of dealing with these extreme microbes. And so, if we're out of drugs, we actually may go back to developing phage-based antimicrobials for combating some of these. And if you're interested, take a look at that article in the March issue of Microbes on phage and phage therapy. It's an interesting read.
It's really interesting, Michael, that you bring this up, and I think we touched on it before, but if you would have brought up phage antibacterial therapy 15 years ago, you would have been laughed out of a room because nobody would have considered it. But right now, phage therapy for at least wound infections and other topical topical agents applications is now very seriously being considered because, as you say, we're kind of out of new technologies to deal with a growing problem.
And the physicians are rightly concerned about this. I always point out to individuals that the microbes gave us about 100 years worth of oil, and they've given us 100 years worth of antimicrobials. And what they have been trying to teach us these past 200 years is that you have to get your energy house in order, and you have to figure out how to easily coexist with us, because while we've given you antibiotics to allow you to figure us out, you're really going to need to figure us out to coexist with us. And the antimicrobials, unfortunately, were a blunt tool to control the growth of microbes when they ended up in an undesirable location.
All right, next up on TWiM, we have a few emails before we wrap it up. And we're getting more and more, which is great. We thank everyone for doing this. The first one is from Stephen, who writes, TWiV, TWIP, and TWiM are wonderful for a retired person like myself. I worked in IT, but started out with a biology undergrad that instilled my interest in biology many years ago. I always look forward to the next podcast. I have discovered that dinner conversation about TWIP is not as well received as a discussion about my latest project from the course I am taking at the College of Craft and Design. He sent the link to the XKCD comics, which many of you may know of, science-based comics. Thanks, Stephen. The next one is from Giselle, who writes, I'm a big fan of all your podcasts. Copper is used as an anti-parasite agent in marine aquariums. It's toxic to all invertebrates and is deadly to them, so it is used in fish-only quarantines. This is perhaps why there is a concern about copper in the marine environment, as it will not only kill microbes, but snails, crabs, squids, etc. All right, that's from our first TWiM on copper. The next one's from Eric, who writes, Hey guys, love your show so far. I have a few thoughts on that last episode concerning Bacillus anthracis, particularly the lack of variance in anthracis. I found it very interesting to learn that spore-forming species have characteristically more genetic variance than non-spore formers. Does the lack of genetic variance have something to do with the fact that anthracis forms endospores and not spores in a fungal sense? Fungal spores are easily released into the air for dissemination, while anthracis endospores seem fit to persist in the soil and dead bodies of infected animals. Do fungal spores show more genetic variation in a strategy to survive in a more diverse environment? Maybe Bacillus anthracis spores are less inclined to disseminate throughout the environment because the cattle and sheep they typically infect die within a few days and are generally confined to the field of the farmer who owns them. Thanks. Keep up the great work. Any thoughts on that one?
That's a hard question, but I would go back to one of the principal attributes of bacterial endospores, and that's the ability to resist heat. Since these are soil microbes, the soil temperature often exceeds the temperature of life. Soils get oftentimes very warm because they're dark and sunlight magnifies the heat, and so the endospore, being able to withstand high temperatures, will allow those microorganisms to effectively survive the high heat of the day, while at the same time then germinating at night, living their life in the dark and in the cool, And then the next morning, when it begins to get warm again, they'll sporulate and live on to fight another day. While the fungal spores typically don't have the same resistance to heat. So I think the bacterial endospore, it's about resistance to those harsh environmental conditions that really drove their genetic selection rather than anything else.
Okay.
And drying, too.
And drying, absolutely, the desiccation part.
Our next one is from James, who writes, "Hello, professors and expert. I am hoping for an upcoming episode on resistant bacteria or upcoming antibiotic drugs in the pipeline. Your fellow cytogenetics friend, James, is from the Northwestern Memorial Hospital." Yes, we shall.
We shall.
I think that'll be a great episode. Next one's from John. John writes, "Dear TWiMers, I love this new podcast, and I especially enjoyed this episode." He's referring to the one where we talked about the FBI investigation. "This is a nice example of both the strength and weaknesses of scientific forensic evidence, I think. You can get useful information out of it. The anthrax strains in Ivan's lab are very likely the source of the anthrax used in the attacks, but not the critical questions you care about. Was it Ivans who made the spores and mailed them, or was it someone else? If it was Ivans, did he have any help? I suppose we will never really know for sure since he's dead and the FBI's investigation is over. Assuming Ivans was the attacker, this raises an interesting point with respect to the threat of bioterrorism. Ivans was a U.S. government biological weapons researcher working on defending against a potential attack. He had expertise, equipment, training, supplies, and access to information at a level that it's hard to imagine any other terrorists having. And yet, his total body count was quite small as these things go. Some nut with a gun in a crowded mall might manage to kill about that many people. The attacks were quite effective at spreading terror, especially among people at the top of government and media, but they weren't actually all that deadly. My question is, does this suggest that bioterrorism is overblown as a threat? Or is it just that this attack wasn't targeted at killing large numbers of people, or that we got lucky and things might have ended up far worse? My understanding is that anthrax isn't really much of a risk to spread after it has been used, and once you know people have been exposed, you can give them antibiotics that will save them, so the attacks were probably self-limiting. Maybe the situation would have been different had the attacker had access to a different agent. So, Ron, this is the one I had sent you a while ago, and since you're here, you can answer it in person.
Well, in fact, the real fear is that someone next time won't announce that they have sent spores of anthrax. Anthrax spores, if spread as an aerosol over a community, can have a very high fatality rate before one realizes that you've been attacked. But indeed, the point that is raised by John is an important one, and that is, no one's really sure whether or not biological agents are great weapons. Certainly, military organizations had major programs in forming these, and they've been given up. So, terrorists, I think, are more interested in fear. And if we go back to 2001, we certainly were afraid of white powders and possible exposure to anthrax spores.
Yeah.
Vincent, wouldn't it make more sense if it was more of a viral agent? That's really the right. I mean, you're the virologist, so for example, someone got their hands on those precious vials of smallpox, right? And you knew how to do it. You inoculated 40, 50, 60, people, and then they got on a plane and then they disseminated them. By the time they were fully infectious, they were exposed to as many people as possible. Then you may have a problem, right?
Well, in fact, all you need is one person with smallpox infection. If they walked into New York City, they could infect quite a few people.
Right.
In fact, that's what happened in the 1940s when there wasn't much smallpox in the U.S. And a man came from Mexico, I believe. He had smallpox, and he infected quite a few people in New York City.
Right.
So there are a few viruses that you could use to do that, but it's now we're quite prepared for that, at least for smallpox. Perhaps some other viruses would be effective. Some of the more lethal ones don't transmit well from person to person, right? So
I'm in agreement with the person who sent in the email that as Ron suggested, we put a lot of money into them, and
I think that Ron is right. The fear is the big thing.
Right.
And how many people die, maybe not so important. It's an interesting topic, though. The next one is from Jim, who sends us a link to a Japanese site. He says it's a good source of information about Japanese activities, has links to seminars, abstracts, software information that's being used, project background, 3D visualizations, and progress reports, all in English. Might be of interest to listeners who wish to see what's going on in the East. And this is a site, Structural Biological Wholesale Project, on T. thermophilus, all sorts of things that are going on
Really?
out there. Yeah. So thanks for that, Jim. Next one is from Kerry, who writes, there's an effort underway to ban triclosan. And I would like to hear TWiM discuss the risk-benefit to using antibacterial agents, particularly their widespread use in consumer products. Any info on how useful triclosan is, whether we'd miss it if it was banned, would be appreciated. Kerry is a loyal layman listener to TWIV, TWIP, and TWiM. I think that's a good topic for a future show, don't we think so?
That would be a very interesting discussion, especially if you asked for a guest host in the form of Stuart Levy. He has strong opinions on the use of the wholesale use of antimicrobials like triclosan, and I think he would be a great adjunct to comment on the triclosan debate, as well as antibiotic use in general.
Okay, we'll do it. I'm sure he'd love to come on TWiM. Our next one is from Clyde, who writes, I just wanted to comment on your short discussion of nomenclature for Salmonella. Panama is not a serovar of Enteritidis. Salmonella is currently recognized as having two distinct species, Salmonella enterica and Salmonella bangori. Salmonella enterica has 6 subspecies, of which subspecies enterica is the most clinically relevant. Within Salmonella enterica subspecies enterica, there are many serovars, of which Panama is a representative. Enteritidis is also a serovar of Salmonella enterica subspecies enterica. Therefore, the proper name for this outbreak strain would be Salmonella enterica subspecies enterica serovar Panama. Whenever Salmonella Panama is used, or Salmonella enteritidis, it implies that the serovar is a member of the Enterica subspecies Enterica family. Wow, Clyde, thank you. And I couldn't be more confused.
As my old advisor used to say, God save us from the taxonomists.
I was going to say, I love the taxonomists. They keep all of us, or we bacteriologists, honest.
All right, let's do one more, and then we will wrap this up. The next one is from Nathan. To Vincent and everyone on TWiM, I have thoroughly enjoyed these podcasts. They are perfect entertainment while I am stuck spinning down large culture volumes. I especially enjoy the format. Focusing on just a few papers, topics really allows for in-depth discussion. Please continue your coverage on all the human microbiome work. It is a fascinating topic. As an undergrad at Cornell, I was lucky enough to take the first offering of a course devoted solely to our microbiota, especially its positive impact. The course sold me on the importance of this field and its potential to change the way we look at some of the most important diseases that afflict us today. Also, I'm looking forward to your coverage of bacteriophages and phage-related therapy, especially the future of its use in medicine. You know, a course on our microbiota, that sounds great.
Can we sign up?
Yeah, we should find the person who taught it at Cornell.
When you and I were there, Cliff, they certainly didn't have that.
No, I know, but they didn't. But we should invite the person who taught the course. Because
But when you were there, was the most popular course at Cornell winemaking?
Absolutely. And you know what? Everybody wanted to take that course.
It probably still is.
Well, that and beekeeping. I took beekeeping because it would look better on my transcripts rather than wine tasting.
Oh, you didn't take wine tasting, huh? Yeah,
no,
wine
because,
tasting.
Yeah. Don't you remember everybody was signing up for it?
Right. Okay, that'll do it for another TWiM. You can find us at microbeworld.org/TWiM. We're, of course, on iTunes and at the Zune Marketplace. And you can subscribe at those sites so you can automatically get each new episode. We also have an app to listen to TWiM on your iPhone or Android device. You can find it at microbeworld.org/TWiM. Please send us your questions and comments. You heard them today. We really enjoy answering them, send them to twim@twiv.tv or go over to microbeworld.org/twim, where you can also leave a comment. And as I said earlier, if you have a story you'd like us to discuss, go over to microbeworld.org and tag it with TWiM. I'd like to thank everybody for participating today. Ron Atlas, thanks for joining us at the last minute.
It's a good time to do it.
You bet. It's always good not to plan, right?
My story.
Ron is at the University of Louisville, where the weather's not so good. Cliff Mintz is down in Princeton, New Jersey. Thank you, Cliff.
You're welcome, Vincent. It was a pleasure, as always.
And Cliff is the man who runs biojobblog.com. If you haven't been there, check it out. And Michael Schmidt, the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent. It's been fun again.
And I got South Carolina right this time.
That's right.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for supporting TWiM, Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for organizational and technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.